morning. Okay, so today what we're going to do is start by continuing on from yesterday's lecture, and then we'll go into population genetics of rapidly evolving populations. So just to remind you of where we were, okay, just to remind you of where we were considering these simple models where you have directional selection, mutations and genetic drift in some population of a constant size, and we were asking two main questions, okay, which is how fast does the population adapt or decline in fitness? Okay, and then the second question was which mutations fix, right? So basically, if we were looking at an experiment or some microbial population, we want to know, given some distribution of fitness effects of new mutations, some mutation rate, and so on, how does the evolutionary process itself sort of pick and choose among them to determine which mutations fix, and how quickly the population either increases or decreases in fitness? It'll do one or the other depending on what parameter regime that we're in. So if you remember from last time, okay, we started talking about, we started by talking about a single S model, where we only had beneficial mutations of some characteristic fitness effect S, and we talked about the traveling wave in this sort of discrete space where individuals had some discrete number of mutations, and we looked at what the shape of the wave was, and we showed the answer to this first question, and we showed that when mutations are rare, the rate of adaptation, meaning the change with respect, the change per unit time in fitness in mean fitness of the population, would scale linearly with the population size and mutation rate if mutations were sufficiently rare. And when mutations are not, which as we argued would be the case in essentially any reasonable microbial evolution experiment other than those specifically designed for the purpose of being in this regime, we found that we got this very different result. Okay, which is logarithmic in the population size and mutation rate, basically a much, much slower rate of adaptation than we would have got if there was a purely sexual population, if we didn't have to worry about these interference effects. And this, as we mentioned, was one of the reasons for the evolution, one potential explanation for why sex confers an evolutionary benefit. But within this model, we couldn't really talk about which mutations fix because there's only one kind of mutation. So of course, all the mutations that fix are of this type. And so then we started thinking about a more complicated model where we still have, just directional selection. But now we have mutations that can confer some distribution of fitness effects, right? And so we defined this distribution of fitness effects rho of S. This is basically describes the probability that a given mutation has a given fitness effect. And we could imagine that this includes both beneficial, deleterious, as well as neutral mutations. And so now we can start to ask this second question, okay? And where we finished up last time is, I'm going to just move over here so as not to draw across this line, all right? We show that we still get a sort of traveling wave of fitness in this situation, okay, where there is a roughly Gaussian distribution of fitness within the population. Okay. okay, which moves towards the right or to the left, depending on whether there are, you know, a lot of beneficial mutations or no beneficial mutations or whatever, but it'll move to the right or to the left with some characteristic V, similar to the single S case, and it will maintain variation in fitness because of this balance between mutations creating new variation and selection killing the guys on this side and expanding the guys on this side in such a way as to maintain a roughly Gaussian distribution of fitness with some standard deviation equal to the rate of adaptation or fitness loss, right? Or more precisely, V equals sigma squared plus the mutation rate times the average effect of a mutation, okay? Which is a sort of just a slight generalization of Fisher's fundamental theorem, which is important when mutation rates become substantial, okay? All right, so that's where we wound up, and then we were sort of we were talking about how in a in order to answer the second question, what we would have to do is think about mutations coming from different parts of this distribution and landing in different parts of this distribution. So, you know, we sort of mentioned sort of briefly this balance between there are a relatively large number of individuals in this blue part of the fitness distribution, which is a little bit further from the front relative to the number of individuals there are in this part of the fitness distribution. These are the sort of really extremely lucky genetic backgrounds that are most fit in the population. These are sort of better than average, but not dramatically so, fitness backgrounds that are maybe 1 or 2 standard deviations above the mean. And because there are more of these individuals, there are more mutations coming from these individuals, right? And so, under the sort of simple assumption that I'm taking a fairly agnostic view to what this is, but we are going to assume that large effect mutations, large, strongly beneficial mutations, are rarer than weakly beneficial mutations. And so in these in this situation, this mutations from this class of individuals, there are more of them than mutations from this class. So we're more likely to sample a large effect mutation from this to land somewhere out here in the front. On the other hand, mutations coming from this exceptionally lucky genetic backgrounds don't have to be very good at all to land in the same place. So it's this balance between lucky mutations on middling genetic backgrounds versus average mutations on very lucky genetic backgrounds, okay? And so now we're going to pick up there, and we're going to sort of say something at least a little bit more quantitative about it. And I can't really sort of derive this on the board in any kind of really precise way, just because it would take 2 days worth of lectures, and I want to talk about other things as well. But I'm going to give you the sort of basic idea behind the calculation and how it relates to the more detailed calculations that we did yesterday. And if you're interested, I'll also give you the reference to look at in more detail. Okay, several references. All right, so we have this. And so to sort of draw back to the picture that we had last time, when we had a single S model, right, we had this sort of discrete fitness distribution, okay, where we had just defined the mean as having a fitness of 0, just by definition, since we're only worrying about relative fitness. And then we had these discrete classes of individuals. And you remember the basic idea was that we treated the sort of mean fitness, these bulk of this distribution, in some nonlinear but deterministic way, and then we treated the mutations at the nose in this sort of crude, linear but stochastic way, and then we sort of coupled the two. And we coupled the two by demanding, basically, that the rate at which the nose expands has to equal the rate at which the mean advances, right? And so that was our sort of consistency condition that allowed us to couple these 2 different descriptions of the 2 different parts of the distribution and sort of use the theoretical approach that's most appropriate for each part. Okay, over here, it's sort of not nearly as clear how we want to go about doing that, right? And this was what made this problem a lot harder to solve than this problem. It's still fairly straightforward. Okay, we've already effectively calculated what's happening to the bulk of the distribution. We have some approximately Gaussian distribution. It's moving towards higher fitness at this rate. Okay, there's some unknown parameter here, which is the sigma squared, or equivalently the v, all right? And somehow that's the parameter we're going to want to couple to the dynamics here at the front. So how do we want to do that? So the way that a bunch of people have now approached this problem is to ask about using a branching process to ask about the fate of mutations here at the front. Basically, in the long term, one of two things can happen to a mutation that occurs, somewhere here in this forward tail. Okay, it can either fix or not, right? And it can fix. In order for it to fix, it has to do two things. It has to survive genetic drift, and it has to sort of outrun everything else, right? It's not enough to survive genetic drift if you get sort of swallowed up by other things that jump past you. You have to not only survive drift, you have to outcompete everything else and not get sort of leapfrogged by other mutations coming and jumping over your head out forward of you. If we could calculate the probability that a given mutation, okay, were able to fix in the population, right, then what we would be able to say is that the rate of fixation of these mutations, okay, that implies a rate of adaptation. And if you think about it, if a mutation of fitness effect S fixes with some probability, and we know that mutations of fitness effect S occur at a certain rate, we can calculate the sort of time averaged rate of change of fitness in the population. Okay, so we will know. Okay, let's assume, if we could calculate pi of S. It's sort of like what Lindy and Brian have talked about, but now this pi of S is the fixation probability, not the establishment probability, not the probability of surviving drift, but the real fixation probability of some mutation that confers fitness advantage S, okay, that just occurs at random anywhere in the population. Okay. If we could calculate what this guy's fixation probability is, okay, then we could say that the rate of adaptation, okay, equals the rate at which these mutations occur, okay. So this is the rate at which mutations of fitness effect between S and S plus ds occur. The population size times the mutation rate times the probability that the mutation has that fitness effect, okay, times the probability that that mutation fixes, okay, times the fitness effect that it provides, right? Because on average, the rate at which the population increases in fitness, well, it increases in fitness by accumulating fixed mutations over the long term. And so these are the rate the mutations appear. This is a probability it fixes. And obviously, if you fix neutral mutations, you don't care. You also have to multiply by the fitness effect provided by each mutation. Buys you more to fix a large effect than a small effect mutation in terms of change of the fitness. Okay? And we could also say which mutations fix. Okay. Right? If we knew this, we could also say that the distribution of fitness effects that actually fix in the population, okay, would just be proportional to, we have to put the right constant of proportionality so this is normalized, but is proportional to the rate at which mutations of fitness effect occur times the probability that they fix, right? That's fairly trivial, okay? So the key quantity is going to be to compute this probability that a mutation fixes, all right? So how do we do that? Okay. So this is where I'm going to try to give you the basic idea and then wave my hands a little bit. All right. So the basic idea is, for a moment, we're going to step back from fitness effects of mutations and just ask, where does a mutation land? Okay. A mutation lands at some x in this fitness distribution, right? We'll call that the landing fitness, okay? And later today or tomorrow, when we start talking about experiments, we'll talk about how at least very roughly, this is actually a measurable quantity. Like if you have a new mutation, you can actually measure to some extent the landing fitness. It's much harder to actually measure the fitness effect of a mutation than it is to measure the landing fitness, as we'll see. But anyway, we have a mutation, and maybe it's a very strongly beneficial mutation occurring in a middling background, or maybe it's a very weakly beneficial or even deleterious mutation occurring in a very lucky background. Whatever it is, it lands at some x. Okay. Okay. We want to ask, what's the fixation probability? Okay. And we'll talk later in a few minutes about how that relates to this pi of s. Okay. Okay. And we'll call that W of x. Okay. This is notation introduced by Oskar Halicek in a paper that I'm plugging to everyone who will listen in PNAS in 2011 called The Noisy Edge of Traveling Waves, which I think is, one of the most important, if not the most important paper in this field in the past few years. So we want to ask, what is this probability? So first of all, can anyone tell me if x is really large, like way out here on this side of the board, what should W of x be? Well, it will be close to 0, but it's actually about as big as it can be, right? As for really big x, that's a really fit mutant. If you get far enough, it should be 1, I agree. But I'm still talking about x small compared to 1. Okay. So a fitness advantage of 10% or something that might be well past this wave, but still not enormous. Very good. Yeah. So if you get far enough out front, okay, you don't care about everybody else, right? Maybe you go extinct due to genetic drift, then you're toast. But if you survive genetic drift, you're going to fix, because nobody's ever going to catch up to you. You got too big a head start, okay? So we know for large enough x, okay, if we look out here, let me draw in a different color. All right. And actually, I'm going to erase over here so I have a bit more room. Okay, so if we have large enough x, okay, we know that W of x has to be proportional to x. Okay, it's maybe x or 2x depending on what stochastic model we're using. Okay, now over here, what's W of x? I mean, it's got to go to 0, right, if we go far enough left, right? I mean, I don't care how beneficial you are. I mean, if you start out less fit than everybody else, you got a real uphill hill to climb, right? So we know that it's going to go like this, I mean, if you land below the mean fitness, forget about it, right? And somehow, this has to sort of come up and then sort of match this, okay? And in here, like if we were to just draw this sort of back, it should intersect 0, obviously. It should be a straight line here. It's proportional to x. I draw that a little better. Okay. So as we get sort of back here, the fixation probability is depressed relative to what it would be in the absence of anyone else. And that's basically due to the fact that even if you survive genetic drift, which occurs with this probability, there's a good chance that you'll be outcompeted by other things in the population. Okay. Now, I'm going to wave my hands at this point, right? Because without getting into several days worth of math, probably I couldn't really explain in detail. But roughly speaking, actually, there is some sort of critical threshold here, right near the front of the wave, okay, where beyond this threshold, this W of x is approximately x. You've gotten far enough ahead that you're not going to get outcompeted. And below this threshold, this rapidly declines towards 0. Okay? And essentially, most of the mutations that do fix, okay, will be centered around this point. Okay, exactly how broad this is depends on the details of this row of S. But basically what you're seeing, what you're going to see, is that it doesn't pay to try to land too far ahead of here, right? I mean, it's obviously better the further that you land ahead, okay? But you want to, but you know, it's harder to land further ahead, right? You have to get some rarer event of some rare large effect mutation on some exceptionally lucky background. And when that happens, those mutations will fix, but it'll happen rarely. Instead, most of the mutations that fix will be ones that sort of land just far enough to have a reasonably good chance of escaping this sort of effective interference from everybody else, but aren't the ones that jump, you know, way, way out ahead of the mean, because those mutations are really rare. Okay? So we can compute this W of x, okay? And from that, we can get this sort of distribution of these landing probabilities, okay? So with this W of x, we can now calculate this quantity that we want to calculate, right? So if we have W of x now, okay, from W of x, what we can calculate is the fixation probability of a mutation with some arbitrary fitness effect. And the way we can do that is ask, imagine that you have a mutation that has fitness effect S. The question is, where does it land? Well, that obviously depends on what genetic background it occurred in. If you have a mutation of fitness effect S and it lands on this background, then it occurs on this background, it will land here. So if it occurred on background X0, it'll land on background X0 plus S, right? So what we need to do is we need to average over, because it occurs on some random background, we need to average over those backgrounds. So the probability it occurs on background X minus S is F of X minus S, right? Times the probability it fixes. If it occurs on background X minus S, it will land on background, it will land at fitness X, and its probability of fixation will be W of X. And we need to average that over all possible X's that it could land on, right? All possible backgrounds it could land on. Okay, so this gives us the pi of S, okay? The fixation probability of the mutation given its fitness effect, all right? And this basically captures this. Well, actually, I'm not quite there yet. Okay. Right. So from this, we can get, as I said, the rate of adaptation is the rate at which these mutations occur times their fixation probability times their fitness effect. Okay. And the distribution of fitness effects of fixed mutations will become some constant of proportionality times the probability of fixation of a mutation times the probability that mutation occurred in the first place. Okay? And this will now capture this sort of intuitive effect that I described before, right? Mutations, if we think about the mutations that fix, mutations of very large positive effect will be relatively rare. Okay? On the other hand, they will be able to occur on a wider range of genetic backgrounds than, and still land at some reasonably high X. Okay? So basically, this part will be low, but this pi of S will be higher because you'll be averaging over, for a given X, you'll be able to sort of get further back in the distribution where this F is larger. Okay? On the other hand, sort of weakly beneficial or even deleterious mutations, the W of X is really small unless you're at the very, very forward edge of F, at which point F is small. Right? So this is sort of the balance between these two effects. The W is increasing as you go out in this direction, this green line here, and the F is decreasing as you go in this direction. That's this white line. And so the mutations that fix are sort of balancing these two effects. okay? And that's what gets us this. All right. So I'm just going to sketch out what this might look like for a typical distribution, just to give you some sense. Okay. So let's imagine that we had some exponential distribution of fitness effects. Okay, some average fitness effect of a beneficial mutation S0, and rare beneficial mutations. And I'm just imagining no deleterious mutations for the minute. I'll talk about them in just a sec. But okay, we have this minus S over S0, so it's an exponential. All right. So on this scale, I'm just going to draw a log scale. This is the DFE. Okay. Rare, large effect mutations are rare compared to small effect mutations. Now, we wanted to talk about which mutations fix. So in the case where there is no interference, okay, if we had a really small population and the sort of regime we talked about at the beginning of last time, what would pi of S be? Okay, if mutations were rare. Yeah, right. So in that case, we would basically be saying that rho f of S, okay, rare mutations, okay, we have to have a normalization constant, but it's proportional to S rho of S. So really weak effect, almost neutral mutations would have essentially 0 probability of fixing, okay? And then, let's see if I draw this right on the log scale. You'll have some probability. Try to draw that better. Some probability kind of like that, okay? So this is the distribution if mutations are rare. Okay, and then when mutations get common, what will happen is they will actually, we'll fix more like this. Okay. Okay. Because intuitively, what's happening is, okay, rather than this being proportional to S, okay, this is really going to now much more strongly cut off the weakly beneficial mutations, right? Strongly beneficial mutations can still get out, right? If you had an arbitrarily strong, if you had a very strong effect mutation, right, it can occur in any individual and just jump way out here and fix with probability X, okay, with probability its fitness effect. But a weakly beneficial mutation can only occur in some subset of these genetic backgrounds and get to some substantial X. So we will always have this pi of S being less in this clonal interference case than it was in this rare mutations case, okay? And so this effect is the loss of mutations due to genetic drift. I'm sorry, due to interference. Okay? And so what you can see is that what happens is, the sort of stronger the interference is, the further over in this direction this distribution of fixed mutations becomes. And basically what it does is it biases the mutations that fix to be more and more sort of out in the tail, the sort of best mutations that could have occurred, right? Whereas in the rare mutations case, you're sort of much more dominated by which mutations happen to occur. In the common mutations case, you're sort of more consistently getting the best mutations are the ones that fix, right? It sort of makes sense. If you have lots and lots of mutations occurring all the time, then the population is going to more reliably find the best ones. Whereas if mutations are rare, you're just going to kind of fix what what mutations are lucky enough to occur in the first place. Okay? All right. So one other thing that it's worth looking at, just briefly, because this gets to a counterintuitive thing that I was mentioning yesterday. Is if we just look at pi of s, okay? All right. Don't really need the negative part. All right. If mutations were really rare and we just had fixation or loss due to genetic drift, what would pi of s be? Right? Does anyone remember the Kimura formula? Right, Lindy talked about this, I believe. Right? Say again? Yeah. Very good. Okay. This would be what it is in the absence of interference. Okay. So I'm going to draw this in blue. Okay. Okay, so what you would get, something like this. Okay, eventually it would saturate here for very large s, okay, approaching 1. Okay, so basically what would happen is neutral mutations have a fixation probability of 1 over n, right? If you have a neutral mutation, it fixes with probability 1 over the population size. That's going to be always true regardless of interference or anything like that. And if there was none of this interference effects, and it was just all you had to do was sort of survive genetic drift to fix, then basically between minus 1 over n and 1 over n, so fitness effects of ns, absolute value of ns small compared to 1, the fixation probability would be approximately neutral, right? For mutations that had a deleterious fitness effect substantially less than minus 1 over n, they would essentially very quickly exponentially decline in probability of fixing, and very quickly that would go to 0. They would essentially never fix. And then for beneficial mutations with fitness effect much greater than 1 over n, they would fix with a fixation probability proportional to their fitness effect, right? That's what we saw from this. So for interference, what happens is, okay, still neutral mutations fixed with probability 1 over n. Nothing has changed about that. But essentially now this regime of sort of almost neutrality is extended further out, okay? Because mutations that have some fitness effect that's maybe much greater than 1 over n still are so small that they routinely get leapfrogged by bigger effect things, okay? And so they don't really fix. And so this sort of will be extended out further, and eventually for large effect enough mutations, these mutations are very rarely leapfrogged, and so it will go back to being proportional to n, okay? And eventually saturate at 1, of course. Now on this other side, okay, what's going to happen? So this I didn't really talk about intuitively in terms of this distribution, right? But in this rare mutation limit, deleterious mutations always land below the mean fitness, right? Because everybody has the same fitness at any given time. Mutations sort of occur rarely. And so every deleterious mutation sort of sends you back below the mean, okay? But now, when this whole distribution of fitnesses, it's rare, but occasionally a deleterious mutation can happen on a really good genetic background. And it'll land at some x where it'll never land like way out in the front where its fixation probability is proportional to x, but it can occasionally land in that sort of shoulder region where the fixation probability is not 0, okay? And so this will actually extend this regime of effective neutrality further back, okay? Of course, deleterious mutations can never fix more often than neutral ones, right? So this can never go above 1 over n. Okay? But it stays close to 1 over n until we get far enough back. And so how far back do we have to go? Anyone give me some rough intuition? Like, what does it depend on? Sorry, somebody think? Yeah, it depends on n. In terms of this distribution that I talked about, the distribution of fitness within the population. It'll depend more on how much variation there is, right? If the distribution is really wide, right, then there are a reasonable number of individuals that are, substantially more fit than the mean, right? If the distribution is really narrow, let me put it this way. If the distribution is really wide, you can occur in a very lucky genetic background and go back by a given effect and still be fairly lucky, right? If the fitness distribution is really narrow, then you can't go back by much and not get below the average fitness, right? Okay, maybe sketch this, right? Let's imagine we have a narrow distribution versus a wide distribution. Sorry, I meant to have the same mean. Okay, imagine you have a deleterious mutation with a fitness cost of some amount, right? Here, if you have a fitness cost of, say, this much, okay, you can really only occur in this background and still land somewhere above the mean, right? On the other hand, for that same fitness cost, you could land on all of these genetic backgrounds and still land above the mean. And moreover, you could have a much bigger fitness cost and still land in sort of the same proportion of backgrounds and still be, the same relative distance above the mean. So basically, the upshot is, this depends on how much variance there is in the population, sigma squared, or alternatively, v, the rate of adaptation in the population. And as sigma squared gets bigger, v gets bigger. So the faster the population is adapting, the further back this will extend, right? This is the basis, and eventually it will go to 0, of course, same as before, right? But this is the basis of that intuitive, sort of counterintuitive thing I was saying yesterday, which is that as the population adapts faster, it has more variation in fitness, and therefore more strongly deleterious mutations can fix, right? So sort of counter to the intuition that you have from the single locus regime, as you increase the population size, right? As you increase the population size, these sort of become narrower together, okay? And so you might imagine, based on this formula, that less deleterious mutations will fix. Actually, the opposite is going to happen. As you increase the population size, v and sigma squared are going to go up, and this is actually going to extend further back, right? Well, that's like one sort of counterintuitive and testable prediction of these kinds of thinking, all right? Okay. Yes, and let me just say, so they're very complicated expressions which involve sort of they're sort of implicit expressions, sort of like the expression for Q last time where we had, Q on both sides of the equation and we could get approximations. There's sort of even more complicated versions of that are known. And I'll just, for those of you who are interested in looking further, there are a few papers that I recommend. So I guess 2011, I think. Okay, so this is Oscar Halachek's paper, which sort of set up this whole framework. This is the paper by a graduate student in my group, Benjamin Good, which works out these formulas. And this is a paper by Daniel Fisher, my former thesis advisor, who, I mean, this paper is a chunk to bite off. I mean, sort of, if you're interested in looking at it just for sort of sociological reasons, it's kind of entertaining because it's this like 70-page paper in Journal of Statistical Mechanics. It had no figures, okay, until I convinced him finally to put in at least 1 or 2 figures. There's just 70 pages of solid calculations. I don't actually recommend you read it, but it's sort of the definitive source if you want any sort of aspect of these calculations. Okay. Yes. Yes, and so this I've sort of not really given due discussion of, because as sigma squared gets wider, gets bigger, obviously the sort of f that you can land on expands in a sense. But the w also sort of, you have to land further ahead of the mean in order to fix. So the w doesn't sort of expand in a proportional way, okay? And so those two effects to some extent slightly cancel, okay? But this effect wins. So that's the details of it. But yes, you do have to consider that. No. Certainly you could set up the population. I mean, here's just a really dumb example, right? Like, you could set up the population with the distribution of fitness. You could set up a population of size 10 with some distribution of fitnesses just by picking. And then, you know, if you happen to get a deleterious mutation on the most fit of those guys, it may fix, even though the sort of steady state F in that population would have been such that that could never happen. So it depends on the actual F in the population at the moment, right? And so, yeah, I mean, obviously, on average, in the long term, this will be at the steady state, and that's where these expressions come from. But yeah, if you sort of set it up in an artificial way as to not be that, then you would have to use the actual current F. Okay. Okay. So I want to get at least started on experiments today. So I'm going to just sort of talk briefly about this other sort of theoretical question about population genetics within these populations. So what I mean by this is probably not very good use of terminology. So far, we've talked about what mutations fix, right? How rapidly the population evolves, what mutations fix, okay? And that's sort of like equivalent to thinking about divergence, right? So like if you have two different populations that are evolving independently, we would see these fixed mutations in terms of divergence between the two populations, right? However, we might also be interested in looking at genetic diversity within a given population at a moment in time. And that will include mutations that are going to fix and mutations that aren't, right? And so we've talked about diversity within the population in terms of fitness. That's what the traveling wave is, right? It's the distribution of fitness within a given population. But of course, that doesn't necessarily tell us what the genetic diversity within the population is, what we would see if we sequenced individuals, what sort of polymorphism pi would we see? This is a different pi, the heterozygosity pi. What sort of site frequency spectrum would we see if we were to sequence these populations? What would be the genealogical ancestry of the population, okay? So I just want to take maybe 15 or 20 minutes to talk about the sort of intuitively how we think about this kind of question. Okay, so first of all, before I get into the traveling wave situation of rapidly evolving populations, let's ask about when populations aren't rapidly adapting and beneficial mutations are rare. Okay, I should say, if there are no beneficial mutations at all, right, and we have a population that is just subject to purifying selection, all right, when provided those mutations aren't, you know, too common, in which case we also have a traveling wave of deleterious mutations. But in the case where those aren't too common, they're sort of rare deleterious mutations, are rare enough, then the framework of background selection that Brian described a couple of days ago will apply. When there are rare beneficial mutations, on the other hand, okay, we have essentially occasional selective sweeps. Okay. We can think of that in a couple of contexts. So if we looked at fitness, okay, and the fraction of the population, okay, initially everybody would have the same fitness, some mutation would occur, right? A single mutation would occur, create a population that was a bit more fit, a subpopulation that was a bit more fit. This subpopulation, if it survived drift, would increase in fitness, would increase in frequency, and then before long, before any other mutations occurred, right, the whole population would look like this, right? And if you were sampling a sample of individuals from the population at this time, okay, so this is, say, the present, okay, and this is the recent past over here, okay, and you imagine taking a few individuals from the population at this time and sequencing them, okay, they recently were the sort of subject of an exponential expansion of a single individual that mutated from here to here and then exponentially expanded. When we kind of think about their genealogy, they sort of trace back to that single individual, right? And maybe some neutral mutations occurred during this time period, okay? But what we would expect to see is they're sort of all descended from this single individual or one of the very small subpopulation that existed right after this mutation occurred. And so basically, they have this sort of star-like genealogy, right? They sort of all are equally related to one another. They all are descended from that same individual. And you know, if we sequence these individuals, they might have whatever neutral mutations occurred in this time, but they'd all have different mutations. There would be no mutations which two of these individuals shared and the third didn't. And the amount of neutral mutations that they have would just reflect how long it has been since this sweep, right? The longer ago the sweep was, the more neutral mutations they would have accumulated. Okay? So looking at this on a molar diagram, okay, what this is, if I look at time this way, all right, and I imagine sort of lining up the individuals in the population in this axis, some time goes by, everyone is genetically homogeneous, then some beneficial mutation occurs, and quickly it spreads through the population, right? And if we take, and now all these mutations, all these individuals have this mutation, now we take a sample of individuals sequenced from this time point, they essentially all descend from this individual. Okay. Now, typically, if we were thinking about a sexual population, we might also have some individual, okay, that we sampled from the present that occurred here, and then there was a recombination event, okay, which sort of separated this individual from this sweep, okay, and we would get some genealogy that looks like this, okay. And that, you know, various folks have talked about, but the basic idea is that what would happen is if we imagined, you know, some set of genomes in the population, let's say we had some genetic diversity looking like this, let's say, in the population, and say some beneficial mutation occurred in this individual here, okay, then over time, okay, this genotype would increase in frequency, all the others would decrease, right? And over time, what we would get is everyone would have this beneficial mutation, and if there were no recombination like we've been assuming, everyone would also have these two mutations here from the background of that mutation, okay? And if there was recombination, then maybe, you know, this individual recombined with somebody that didn't, and so you got an individual that has maybe this mutation and this mutation, but not this one. Okay, and that's what I mean by this kind of picture. All right, I don't want to get into this in too much detail. I just want to say, okay, what's going to happen now when we start thinking about what the genetic diversity will look like in these sort of rapidly evolving populations, okay? Okay, and we'll think about the purely asexual case. Then I'll just very briefly say what would happen if you had some recombination. Yes? Old. Yeah. Sure. I mean, you know, there are clear examples of selective sweeps in Drosophila, for instance. I mean, I don't know. Brian can answer this question better than I can, how sort of general these models of selective sweeps are. I mean, there's certainly there's certainly signatures of things that look like selective sweeps in sort of large eukaryotic sexual organisms, like Drosophila. You know, to some extent, it's, you know, hard, for instance, sometimes to distinguish selective sweeps from background selection from, you know, various other complicating factors. And so, you know, I mean, there's a whole literature on this, I suppose, but. Right. 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 But so the tricky thing is, okay, so for microbial populations, right, that are recombining very rarely, or certainly for the experimental populations I'll tell you about in a little bit, you are definitely not getting clean selective sweeps ever, right? I mean, it's always in this sort of rapidly evolving limit that I'm going to talk about. In, you know, Drosophila or humans, for example, it's more complicated because certainly if you just sort of plug in the sort of raw parameters in a naive way and forget about recombination, you would be in this rapidly evolving population limit. Okay. Well, the thing I haven't told you about, I was going to sort of mention briefly at the end, is that if there is recombination, right, as there obviously is in sexual organisms, then we don't really care about the whole genome, right? And the mutation rate genome-wide is not really the relevant quantity. Rather, what's relevant is how many mutations there are within a sort of genomic region that's unlikely to recombine in a coalescence time. And if there is recombination on that time, then those two sort of behave in a sexual way, in a sort of freely recombining way, which, as well, I've erased it now, but as we've said, sort of that goes back to the rare mutation limit, right? So imagine that you had a lot of mutations, but they were all sort of so well separated that they basically are always recombining with each other. Then they sort of behave independently, and we don't care about any of this stuff. Okay. On the other hand, if the mutation density relative to the recombination rate is sufficiently high, then we would still be in these regions, in this regime. And you know, I think it's probably likely that in organisms such as humans, there's some regions of the genome where recombination rates are high and mutation rates, or at least selected mutation rates to mutations under selection are low, and you're in this sort of rare mutation limits, and there are probably other regions where you're not, and where you have to worry about these kind of effects, but that's a whole much more complicated subject, which is certainly not worked out. Okay, so let me just talk briefly about this. Okay, diversity in rapidly evolving populations. So we can think about the same sort of deal in that I drew here, and now when we have a distribution of fitness within the population. And let's just think, so I can at least do something a little bit concrete, not that concrete. Go back to this case of a single S model. There's one selective effect S, and so we have these discrete fitness distribution, right? Okay. Before we talked about, okay, mutations occur, create individuals that are more fit, right? If there was only one mutation that occurred to create this new class, then what would happen is eventually this mutation, if it survived drift, would increase in frequency, take over the whole population, and that would sort of be like an effective selective sweep, right? This population would be genetically homogeneous. There would be then, you know, these individuals would have whatever mutation created this population plus one more, right? These individuals would all have these two mutations plus one more, and so on, right? And things would be simple. But that is actually not the case. All right? And just to give you a sort of sense of what's going on here, we'll do another calculation analogous to what we did yesterday. So we said that this class at the front, okay, for some distribution with width q here, all right, is growing exponentially at this rate. And it's creating mutations here at a rate u times its population size. Okay? And those mutations survive genetic drift with probability QS. So let's ask about, how many mutations are there that generate each of these sort of classes at the front? And what is their sort of frequency distribution? Is there sort of one mutation that occurs and takes over this population, or there are lots of mutations of similar frequency that will form up these populations? Okay. So in order to ask that, okay, we're looking only at mutations that will survive genetic drift, okay? And so I'm going to say that the kth one of these mutations, okay, the first one occurs when this equals 1, the second one will occur when this integral equals 2 and 3 and so on, right? Because this is sort of the time until you get the kth one of these mutations on average, right? The same idea, we did the same calculation before for k equals 1. Okay. Now we're doing it to just look at how many other mutations there are. Okay. So we just plug this in. We get u times e to the qst integrated from 0 to tau k equals k, right? All right. This is dt. Okay. All right, so we get u over qs, all right? Oh, yeah, that's right. Times e to the qs tau k minus 1 equals k, okay? This 1 is going to be negligible, like before, and so we get tau k equals 1 over qs times the natural log of k q s over u. Okay, so this is when the first, the second, the third, etc. mutations occur. And let's ask, all right, I'm going to define some mk. This is the number of individuals in that class that will be in this sort of new class of individuals at this front that will be descended from mutation k. Okay? And I'm going to compare that number to the number coming from the first mutation. Right? If this number is small, then each of these classes is genetically homogeneous. Right? We just have a single mutation here and a single mutation here and so on. But if this number is not so small, then we have multiple different mutations in each of these classes, and there'll be more diversity within the population. Okay? So we can just calculate this. Okay? What is this? Well, these guys grow like 1 over qs, e to the qst, but they start at time tk, tau k. Right? So they're growing exponentially at rate qs. Okay? But each of them started growing at the time at which they occurred, which was time tau k. Sorry, that's a little hard to read. Okay? We can just compute this. Okay? Okay? Just doing that. Now I just plug in this number that I get. Well, what do I get? Okay. Now, I've been a bit fast and loose with various factors of q versus q plus 1 versus q minus 1 and so on, which you can look at, at the papers that I suggested to get sort of the details on how to get that exactly right. But the upshot is, the first class has, if the first class has first mutation creates a class of size 1. Second is half that. Third is a third of that, and so on. Okay. So there's really actually quite a lot of diversity in as each of these new classes is founded. There are lots and lots of distinct mutations that will make up this group of individuals. And, one of them will be bigger than the others, okay, but the first one is twice the size of the second one and only 3 times the size of the third and 4 times the size of the fourth, and so on. So there's actually quite a lot of these. Okay. And so basically what we can do to try to figure out the genetic diversity within these populations is, so now each of these mutations, right, they will sort of start to grow up, all right? So this class will grow, and new mutations are occurring from here, right? And the mutations are most likely to occur in this green background, okay? But they're also reasonably likely to occur in this orange or blue or the other ones that I haven't drawn, right? And so this will these new mutations will subsample from among these, right? And you will get sort of subsampling of this diversity as we go forward, even as you create the new diversity of the new mutations in this class. And this process will continue on and on. Okay? All right. So I'm not going to tell you much more about that, but the basic idea is, okay, there was some other set of different mutations that were the mutations that founded this class from this one. Okay, maybe there was one here and one here, right? A whole bunch of these. Okay. All right, and maybe you got mutations in 3 of these 4, so one of these mutations will eventually go extinct, but then you created this new diversity of these new mutations. And if we sample individuals from the population, basically, let's say we sampled a couple of individuals from here, and we sequence them, what does their genealogy look like? Well, these guys descend from mutants and less fit individuals and back through the distribution until they all come from, the same mutation, let's say. And we can trace the genealogical structure like that by computing these probabilities that 2 individuals from this class came from a mutation in the sort of same lineage in this class. All right. It's sort of very hand-wavy and sort of general, or sort of not, I don't want to take the time to really sort of explain it in great detail. But if you're interested in learning about that in more detail, let me know, and I'll point you to the references. Okay. The same sort of thing happens when you have some general distribution of fitness effects, right? So if instead we had this sort of traveling wave here, okay, we also have a whole bunch of different classes created by distinct mutations. And another way to sort of look at this would be to think about, okay, in the future, this distribution would be more fit. So it would have moved to the right. Okay? And if we imagine sampling individuals and sequencing them from the present, they're sort of descended again, all right, from the forward tail of this distribution, okay? But it's slightly different. Because there are several of these mutations, it's slightly different than the picture that we would get of a selective sweep. And very roughly, if I draw the same sort of Muller diagram, if we think about the front of the population, some mutation occurred here, slightly later mutation occurred here, and so on. Okay. I'm drawing like mutations that occur at one particular time, like in, at the front, and many other mutations are happening. But if I sequence individuals from within the population, right, they will all kind of descend from each of these distinct mutations, and then they will sort of extend back, and there'll be more diversity, and there was some previous set of mutations that had occurred, right, that maybe includes several of these, right? And we trace them back. And so there's a much more complicated genealogical structure in the population. Okay? Thinking backwards in time. Okay. So the paper that I had as sort of one of the assigned readings by Richard Nair is a nice review in, I think, what annual reviews of Ecology and Evolution, one of those journals, that explains these models and these sort of intuitive conclusions in much greater detail. Okay. So I'm going to stop there on all of the math and board, and because I want to shift gears to experiments. But before I do that, any further questions, or while we're starting the video monitor? Evolution experiment. And so it's sort of easy to get bogged down in the math, and I think it's useful to spend a while just sort of thinking about these, these practical experimental things. So I'm going to talk about work, some of it by myself, some of it by many other groups, on looking at the dynamics of sequence evolution, okay, in evolution experiments. And this is a relatively recent thing, so people have been doing experimental evolution since contemporaries of Darwin. Dallinger in the late 1800s was doing experimental evolution in microbes. But obviously he wasn't looking at changes at the DNA sequence level. And now in the past 5 years or so, people have really started to systematically look at individual mutations that occur at the genome sequence level and track them through time and sort of watch these kinds of dynamics that I've been talking about, both at phenotypic levels, fitness levels, as well as the underlying sequence level. All right, so I'm going to just start with a bit of background. First of all, like, why would you want to do experimental evolution in the first place? It certainly has a lot of really basic drawbacks. So, I mean, what we actually care about in reality is, adaptation in nature, right? This is Darwin's finches, and, these populations, of course, they adapt in response to some selective pressure. And this is a sort of schematic of the radiation of these finches, where these sort of beak shapes evolved to fill specific ecological niches. And there's a whole bunch of really nice work in evolutionary genetics figuring out why these different beak shapes are adaptive for the finches with different lifestyles, and actually mapping these changes in beak shapes to specific mutations in specific genes, and understanding what those mutations, what the consequences of those mutations are in terms of the development of these beaks and why those mutations lead to these different beak shapes. You can say a huge amount about this kind of stuff nowadays. And there's some really cool work on it. But the basic problem with any of this kind of study is, really all you can possibly hope to do is sort of some sort of retrospective analysis. You can sort of say what happened. It's much, much harder to say why did this happen and not something else, right? If you were to just repeat this whole radiation, would you get the same phenotypes? If you did, would those phenotypes be driven by the same mutations? Would there be totally different mutations? How deterministic is this process at both phenotypic levels, at genetic levels? In some cases in nature, we have nice sort of serendipitous examples where we can look at sort of roughly similar radiations that happen multiple times. But in general, this is pretty hard to do. So the beautiful thing about experimental evolution is you can maintain now thousands of independent replicate populations in the lab where you know all of the details of the population dynamics, you know the starting genotypes, you know that the selective pressures are identical between replicate lines, and you can just directly ask these kinds of questions, all right? So what do we do? There are basically two types of experiments. So a few of the other lecturers have touched on this. I just want to briefly summarize so we're on the same page. There's sort of two basic schemes for experimental evolution. So on the left here is a chemostat. The idea is you have a bucket full of media. You got a bunch of microbes in there, or yeast, or E. coli, or whatever it is. And you flow in some sterile media. Okay, there's usually some stir to sort of keep this well mixed. And you flow out some sterile media, some spent media here, which contains some cells in it, right? And so what happens in this kind of setup is, right, the microbes reach some stable density within this media okay, which is set by both the rate at which, by basically the inflow and the outflow rates, right? So as new media flows in and old cells are expunged, these cells divide to sort of replace the cells that are moved out, and they stay at some, at least roughly, constant density, and they sort of accumulate mutations that allow them to sort of better outcompete their neighbors. All right, another scheme, which is most of the experiments that I'll tell you about have been done under, although not all of them, is in this sort of batch culture scheme. Basically what you do is you take a sterile test tube or flask or whatever full of media, you put some cells in, you wait a while, maybe 12 hours, 24 hours, whatever, depending on how masochistic you are, and they grow up and they fill this test tube, and then you dilute them. Basically you sort of take some sample up and you repeat this step, and then you grow again. You just repeat this cycle again and again and again and again and again for weeks or months or years or decades. And the other beautiful thing about this is that you can, for most of these experiments, there is some experimental evolution in flies, for instance, where you can't do this, but in microbes, you can just periodically freeze down the population. And so then you got freezers full of the sort of, as Rich Lenski likes to call it, the frozen fossil record of your experiment. And you can revive ancestral types whenever you like. So you can figure out their phenotypes, you can sequence their genomes. As sequencing technology continues to advance, there'll be more and more we can do with these lines that were frozen down 20, 30 years ago. Okay. So until fairly recently, sort of, you could summarize a lot of these experiments, this is probably still true actually, as what I saw when I watched some evolution, right? Basically, much of this work is fairly exploratory, and that's true of most of what I'll tell you today. And you just watch this evolution happens and you tell a story about it, right? So these are a picture. This is a picture of the 12 experimental lines from Rich Lenski's group that are now almost to 60,000 generations. These are the 12, era -1 through era -6, era +1 through era +6, they're called. And you can kind of see their phenotypic differences, all right? This culture is denser than these other cultures, for example. That turns out to be a really, really interesting story, which I'm not going to talk about, but is really cool. These guys evolved citrate metabolism. The upshot is, though, you watch what happens, you see some stuff. It's kind of hard because, you start the experiment. Like in these experiments, I once showed this to a theorist. He was so excited about seeing these experiments, and good friend of mine. And, I showed him the experimental lines, and he's just like, he looks at it, and it's this sort of cloudy, slightly smelly test tube full of yeast. He's just like, you can't see anything, and yeah, that's kind of the problem. You start with a cloudy test tube full of yeast, and you end with a cloudy test tube full of yeast. So it's sort of hard to say what actually happened. But, historically what one could do is you could look at how competitive fitness changes over time. And I'll explain how you measure this in a minute. The other thing you can do is mark lineages. So for example, this is a figure from a paper by Katie Cow and Gavin Sherlock from a half dozen years ago now, where they started a population with 3 different strains of yeast. 1/3 marked with this green marker, 1/3 marked with this yellow marker, 1/3 marked with this red genetic marker, and track the frequencies of the markers over time. And you see these complicated dynamics like this. Okay. So this is even earlier work looking at microsatellite loci, and as a function of time, these are generation times, 90, 180, 630, 720. Different mutations arise that change the copy number at this microsatellite locus, and this is the frequency distribution of the different copy numbers within the population. And you see these mutants arise. In this case here, there was a mutant that dramatically reduced the copy number. Then it eventually went extinct. Additional mutations arise, and so on. So historically, we've been able to sort of look at these crude things, this sort of phenotypic changes measured either by what the damn thing looks like, what the fitness is. We could look at some levels of underlying genetic changes by looking at marked lineages or sort of single loci within the genome. I talked a bit about fitness. This is going to come up again and again, so I just want to sort of define what this is. one of the nice things about these evolution experiments is that fitness really has a sort of very simple one-dimensional definition in these experiments, which is that, if we go back to this, let's say this picture for a moment, right? The fitness of a clone, okay, relative to the other genotypes in the population, is just that genotype's average tendency to leave more or less offspring than the population average in the next evolution cycle, right? And there may be complicated ecological interactions going on, but the fitness really sort of can be defined just in terms of how many on average offspring you're going to leave into the next evolution cycle. And that's a straightforwardly measurable thing. What you do is you take your evolved strain and you take some labeled reference strain, typically nowadays labeled with some fluorescent marker, and you mix these two. And so you get some test tube full of, in this case, white and yellow yeast, and you measure the ratio. And we can do this now with flow cytometry. You can measure, you can count 50,000 cells and figure out the ratio of yellow to white cells among those 50,000 in 5 or 6 seconds. All right. Then you compete this population in the sort of same environment in which you were doing the evolution, and after a day or two, a couple days of that competition, you measure the ratio again. And the difference in these ratios tells you how many more or less offspring the evolved strain is leaving than the reference strain in each of these cycles. And so that gives you the relative fitness of the evolved strain compared to the reference. And then what we can do is measure all evolved strains relative to the same reference. Okay? And so then we can get the relative fitnesses of everything. Okay? This would fail if there were sort of non-transitive fitness effects where like A is fitter than B, B is fitter than C, and C is fitter than A, things like that. And so to check for those, you can have a bunch of different reference strains. It also becomes more complicated if there are frequency-dependent interactions. These things, as it turns out, seem to be relatively rare. Non-transitivity is extremely rare, and although there are examples of it. And frequency dependence is a little less rare, but still fairly rare in these experiments. So I'll talk about them, in passing at a couple of points. But for the most part, we're going to assume this sort of just a simple frequency-independent, non-transitive, transitive fitness, which is true for most of these experiments. Okay. And so nowadays we can actually sequence these populations. This is a picture of a high-seq. And so, for microbial genomes now, this is really becoming very, very low cost. So, nowadays we routinely get, 20x coverage of 250 different yeast genomes for about $3,000. So like roughly $10 a genome. All right. So what do you see when you do this? So this is a figure from Jeff Barrick's paper. This was one of the first papers to really look at genome dynamics in experimental evolution back in 2009. And Jeff Barrick, he was at the time a postdoc in Rich Lenski's lab. And so he sequenced populations from one of the 12 of Rich Lenski's long-term evolution experiments through about 40,000 generations of evolution. In this particular line, the E. coli evolved a mutator phenotype sometime between 20 and 30,000 generations. So he's mostly looking at the first 20,000 generations of evolution. And what he saw is, in green here, is the fitness of this population as a function of time. They do a kind of lousy job of measuring fitness for sort of historical reasons, but fitness, these bounces around, are just measurement error most likely. So the fitness increases rapidly at first, and then it sort of slows down, but still increases through the 20,000 generations of evolution. So this is what you get in terms of the phenotypic change, the fitness change over time in this population. Rapid initial increase followed by slowing down. And there was just a recent paper a couple months ago that came out in Science, taking this and all the other lines, looking out to 60,000 generations. And actually, they see fitness continues to measurably increase even through 60,000 generations of evolution. Okay? So these populations are not reaching any kind of fitness peak or anything like that. They're just continuing to adapt, even relative to the ancestor, through 60,000 generations. And they sequenced these populations, in this case from 5 time points and then through 20,000 generations, then an additional 40,000 generation time point. One of the surprising things they found here is that the number of mutations increases basically linearly through time, through this 20,000 generations. Then it dramatically increases because the mutator phenotype arises. But these first 20,000 generations, the mutations sort of accumulate more or less linearly through time, right? And that occurs despite the fact that the rate of fitness evolution is slowing down, which is kind of odd, right? I mean, this sort of mutations accumulating steadily through time might be thought of as a signature of neutral evolution, yet, you know, a sort of molecular clock. But yet we clearly know that that's not the case. The fitness is increasing through time. Right, so that's a puzzle that's still really actually not fully worked out. So I don't really have an explanation for that. But what I want to mainly talk about is a question sort of that we've been addressing sort of abstractly through theory, and now we're going to see some more sort of concrete posing of that question. So these are the actual mutations that they found, right? What I'm representing here, this is a figure, another figure from this paper. Each of these circles, concentric circles, is one of the E. coli that they sequenced, from this inner circle being an E. coli from generation 2,000, then 5,000, 10,000, 15,000, and 20,000. So they picked one clone, one individual cell from each of these time points. They sequenced that, and what we're seeing here is the various mutations that they observed in those clones. Sort of, you can think about this as a function of time as the experiment went on. Okay? All right. So how many of you would have guessed this? Right? Is this obvious to you? It's a kind of a funny thing, right? Like, nowadays you submit a paper doing this kind of stuff, and everybody tells you how obvious it is, right? But like, I don't know, I challenge you to predict it in advance, right? I mean, these mutations kind of all make sense. Well, maybe probably not all, but many of these mutations and work since. You know, there's been a lot of work on what these mutations are doing, and there's a sort of nice retrospective analysis of, you know, why these mutations are beneficial. But could you say, just looking at this data from this one population, anybody want to hazard a guess on how similar or different replicate populations would be? There were 11 other lines that weren't sequenced at this point. Would you expect them to have the same mutations? What fraction of the same mutations would you expect them to have and not have, right? So this is the kind of question we want to get at, right? So somehow these mutations that you see in this line are the outgrowth of this kind of process that I've been telling you about in theory over the, you know, earlier part of today as well as yesterday, which is that, you know, there were some mutations of various different fitness effects. They fix with various different probability. These populations are definitely in this clonal interference regime where lots of stuff is going on. There's this traveling wave of fitness and so on. And, you know, these mutations are ones that, you know, either were the strongly beneficial ones that were very likely to occur, or maybe some of them are lucky deleterious mutations, and those we wouldn't expect to see the same ones again. So we want to try to be able to understand this kind of question, all right? And from this single line, like, there's not much more we can do than what we did in the sort of, when we were talking about in the natural population, of just sort of saying some retrospective analysis. But the nice thing is we have now replicate populations, right? For these E. coli populations, we only have 12. And in this paper, they only really fully sequenced the genomes of E. coli from this one population. I'll tell you about more detailed experiments next time. But one thing that they could do to just get a rough sense of this was, without sequencing all the other 11 lines, and they've still not really fully sequenced lots of time points from all the other lines, but even without doing that, what they could do is take these mutations that they found in these other lines and just ask specifically, let's sort of look for those specific mutations in the other 11 lines, right? And that's much easier to do, at least it was at the time, much easier to do that than to sequence the whole genomes of the other lines. Now it would actually be faster and cheaper to just sequence the other genomes, but anyway, time moves on very quickly in this field. So for example, they saw this mutation in NADR, which is a transcriptional regulator, that in each one of the other 11 lines, all 11 of them, that mutation occurred. Okay. So this was 100% present in all 11 independently evolved lines. This pyruvate kinase also, this gene involved in ribose catabolism, also mutated. Not necessarily the same mutation at the nucleotide level, but mutations in this gene or region. Okay. This transcriptional regulator is seen in maybe roughly 2/3 of the lines, and so on down the list, right? Okay. All right. So presumably these guys are the sort of most strongly beneficial things, or maybe they occur with the highest rates, some combination of that, and these things are sort of these things are probably also beneficial, but maybe not so clearly, not so strongly so. And then these mutations, I don't know, maybe they're one-off, neutral, or even deleterious mutations. Sort of hard to say, but somehow these results are a signature of this process, right? You can actually, however, also just go ahead and measure the fitness effects of these mutations by basically reconstructing these mutations all in the sort of same genetic backgrounds or in the genetic backgrounds in which they occurred. This data is just constructing these mutations in the same genetic background. So these numbers have to be taken with a grain of salt because the fitness effects in the background in which they occur might have been different due to epistasis. Nowadays, sort of people are reconstructing things more carefully in the genetic backgrounds in which they occurred. But you can sort of see these fitness effects, right? They're all sort of significantly different from 0. These are in percent. But you can see there's sort of some correspondence between, okay, let's just look. Top A, Pike F, SPO T have very strong fitness advantages. There's SPO T. Pike F is not on here. Sorry, top A is there. Yeah, thank you. Top A is here. All right. On the other hand, this NADR here, which is less of a fitness advantage than this top A, this one is at the top of this list, whereas this guy is sort of much further down. All right. Why is that? All right. Maybe it was just chance. Anyway, so we have all this. This OMP-F is sort of strongly deleterious. I don't know what the hell is going on here. So we have some sort of correspondence between what happens in the experiments and these sort of fitness effects of the individual mutations. But it's a little hard to disentangle. All right? Yeah. Yeah. Right. Of this mutation. Okay? The one that occurred in the line they sequenced. All right? All right. So what we want to understand, okay, is how we sort of get, what the sort of mutation rates and fitness effects were, and how we would like to sort of have a complete explanation of how, given this sort of set of things that could happen, an understanding from the theory of the type that I've been telling you about how this sort of dynamics of the process, this sort of way in which evolution picks and chooses among the mutations according to their fitness effects and the population size and so on, into these set of outcomes that we actually observe, right? And if we understood this well enough, okay, we could look at a few lines and observe these outcomes and infer back something about the possibilities, right? The underlying fitness effects and mutations from this kind of data from many replicate populations. And maybe we could just measure some of these things directly. And then predict, if we were to do additional experiments in different lines, how similar or different those results would be. Okay? And I'm going to tell you about some more experiments tomorrow about really sort of trying to systematically do this, both some from my lab and some from other labs. The key thing I just want to leave you with today is that these are all really statistical questions, right? It's never going to be sort of an all or none thing, right? We're looking at the probabilities that different, we want to compare the relative probabilities that different sorts of mutations fix or are lost, or the probability distribution of certain amounts of diversity within the population. And so, although Rich Lenski's lines are really fantastic in that, you have the longest term evolution experiment you are likely to get for some time, 60,000 generations now, there's really only 12 replicate lines, and so there's a lot of questions you can't ask, right? But nowadays, this wasn't possible when Rich started his lines, but nowadays it's possible to maintain thousands and thousands of lines in parallel. Like in my lab, we routinely maintain 5,000 lines in parallel using liquid handling robots to pipette things around and do all these dilutions in these kind of robots. We can measure fitnesses in thousands of replicates at a time in a similar way using high throughput flow cytometry. We can measure 50,000 cells for fluorescence in 800 replicate populations in 2 hours nowadays in our core facility. And we can now do higher and higher throughput sequencing, although that still remains the bottleneck. And so I'll tell you tomorrow about some experiments that sort of take advantage of this throughput and try to address some of these statistical questions in more detail. That's where we'll pick up tomorrow. All right, coffee?